Optimization of a Western blot-based extracellular signal-regulated kinase activation assay to detect sphingosine 1-phosphate receptor 1 activation in mammalian cells. G-protein coupled receptors are the target of more than 30% of the approved drugs in use. Therefore, assays to determine the activation of G-protein coupled receptors are of high biomedical value, as they can be used to determine potential drug candidates. This research is focused on the development of a Western blot-based ERK activation assay to detect the human S1PR1 activation. The focus of this poster is the assay optimization, which is the initial part of the experiment. The assay is developed using C6 rat glioma cells engineered to overexpress human S1PR1 with a green fluorescent protein tag. Wild-type C6 rat glioma cells were used as the control in the experiments. First of all, these cultured rat cells need to be confirmed for the overexpression of the human S1PR1 receptor. This was carried out in two ways. Protein level confirmation was achieved by observing the cells under the fluorescence microscope at 509 nanometers to detect green fluorescence. As seen in figure 1, clear green fluorescence can be observed in C6S1PR1 cells compared to C6 wild-type cells. This is because the C6S1PR1 cells have the GFP tag along with S1PR1 receptors that are expressed all over the plasma membrane. The final confirmation of the receptor overexpression must be done using reverse transcription PCR with human S1PR1 specific primers. This procedure needs to be further optimized. To optimize SDS page, first cells were stimulated with FBS. FBS stimulation activates ERK, which is the target protein in the experiment. After extracting the total cellular proteins, they were size fractionated in SDS page and were subjected to Kumasi staining to detect whether the desired band separation can be achieved. Figure 2 illustrates the Kumasi staining of proteins separated in 12% resolving gel. It confirms that the proteins have been size fractionated as intended. Hence, 12% resolving gel composition was finalized to be used in the assay development. Optimization of the antibody detection is the immediate next step of the experiment. Subsequently, experiments for the time course determination of S1P-induced ERK activation via S1PR1 and the development of the dose response curve are intended to be completed.